Welcome. This is Kcse 2013 Biology Paper 3. Let's see what you are supposed to do. In question 1, the photograph below shows the inner surface of the upper left side of the rib cage. So you can see this is the rib cage, these are the ribs. Then you are told, name the bone covered by the fatty tissue labeled K. Name the bone. This one now, this is the bone that is covered by the fatty tissue labeled K. Now that is the sternum. Then you are told, explain the role of the part labeled M in inhalation. Part labeled M, these are the ribs. M is the rib. Now you are told to explain the role during inhalation. You will say, after the internal intercostal muscle relax, after the internal let's write here this question Roman 2. After the internal intercostal muscles relaxes, the rib cage moves upwards and outwards. After this one increases the temperature and decreases the pressure. This increases the volume, not the temperature, sorry. Increases the volume and decreases the pressure. The pressure in the rib cage. The rib cage, we have said that is the rib cage M. Now after the pressure and the volume the pressure has decreased and the volume have increased, now the air is forced into the lung. The air is forced into the lungs. That is the role of the part labeled M. Now question B, you are told the photograph the mammalian vertebrae. Now you should be able to identify the mammalian vertebrae. This is the thoracic because of the long neural spine and the centrum, which is broad wide neural canal, and the transverse process. Now this is the thoracic vertebrae. Now you are told, state the view of the vertebra presented. Now this is the dorsal view or the anterior view. The anterior should be able to know the difference between anterior and posterior. As in the early video we said, the posterior, this is the downwards, this is the lower part, while the anterior, this is the upper part. Should be able to know the differences. Now this is the anterior view or the dorsal view. The anterior is seeing the upper part, we are not seeing the lower part. This is now the anterior view or the dorsal view. Then Roman 2, you are told name and state one function of the part labeled T. Part labeled T is the neural canal. This is it is pointing the center neural canal. That is the neural canal. Now you say name is this is the neural canal, and therefore this allows for the passage of the spinal cord Yeah. Then you are told Roman 3, how are the parts labeled S and V adapted to their functions? Now S and V, S is the neural spine, and then V, this is the centrum. Now the adaptation of these two, you will say S being the neural spine is long, that is how it is adapted. Now function is to provide surface for muscle attachment. Yes. Now and then in V, which is the centrum, it is thick. That is how it is adapted. It is thick or it is broad to bear the weight of the body. Now question 2. You are provided with a food sample labeled C. Using the reagent provided, carry out the test and identify the food substance present in the sample. Now our space is very small, but we have big space. Now you are required to test for reducing sugar. Now in the procedure will say put 2 centimeters cubed or 2 millimeter of the food substance C in a test tube, and then add equal amount of Benedict solution, and then heat to boil. Either in a water bath, you can write in a hot water bath, or heat to boil. Now the observation were the blue color of Benedict's solution remain blue. It was the observation. Now in conclusion, there was absence of reducing sugar. Reducing sugars are absent. Now we are testing for non-reducing sugar. In this, the procedure is very big. You should be very careful while writing this procedure. Now we'll start saying put 2 ml of C in a test tube, and then add few drops of dilute hydrochloric acid, add few drops of dilute hydrochloric acid, then you place the test tube, in hot water in a hot water bath for about 3 minutes. 3 minutes, and then remove the test tube and cool with cold water. Then cool with cold water. Then add sodium hydrogen carbonate dropwise, drop by drop, until fizzing stops. Till fizzing stops. Then add Benedict solution. Then heat to boil or warm in a water bath. Then heat to boil. Now here the color changed to yellow, then or orange or brown. That is the color you expected. Now conclusion, the reducing, the non-reducing present, and reducing sugars present. Now for procedure 3, for test for proteins. Now you are required to put 2 ml of the food substance in a test tube. Then add sodium hydroxide equal amount and shake. Then you shake. Then add copper 2 sulfate drop by drop. Then shake. Then you observe the color. Here the color changed. Color changed to purple. Therefore, proteins were present. Question 3. Below are some photographs showing some observable features of leaves. Then you are instructed by being told, using the features in the order given below, construct a dichotomous key that can be used to identify the specimens. Now the first one, you should start with leaf simple or leaf compound, then leaf venation, margin, arrangement of leaves, pinnate or trifoliate. Now let's start with the first one, leaf simple and leaf compound. You should be able to identify the leaf simples and leaf compound. So this is the leaf compound, then this is leaf compound, and even this one, this is compound, and there this is simple. Now let's start by constructing. You say 1A leaf simple, and then B leaf compound. Then we start with the first one, the leaf simple. We have said there are 3. Now you cannot write all the names. You should now look for a characteristics that will differentiate the 3. Now you will say here, because you don't have any only one, there are many, you write go to 2, and then you go to 2a. Now you look the 3, how you can differentiate an observable feature. Now I can see parallel veined leaf, network veined leaves. I will say leaf with network veins. Now you say leaf net veined, that is a short form, net veined, and then B, leaf with parallel veins. Now with the network veins there are 2, but with parallel veins is 1. Now we write this one, we write the name of the leaf, which is the Comelinaceae. That is a scientific name, Comelinaceae. Now here, because there are 2, you say go to 3. Then you differentiate. I can see the leaf with smooth margin and leaf with serrated margin. Now you have differentiate them. 3a, you say leaf with serrated margin, and then B, leaf with smooth margin. Then you identify there are only 2, the Malvaceae and the Nystaginae. Now you write their names. Here this is the Malvaceae, and here this is the Nystaginaceae. Then we have finished with the simple leaf. Now we go back to the A, 1b. Go back to 1b, which is compound leaf. Now we have finished with the leaf simple. Now here we say go to 4. Then we write 4a. Let's see the compound leaves. Now here we will say leaf with leaf opposite, that have the leaves opposite, and the other one with alternate. We'll say leaf opposite and leaf alternate. These are the opposite and these are alternating. I will say even this is opposite and this is alternating. Only one is alternating, which one is this one, but these 2 are opposite. I will say leaves with leaf opposite, and B, leaf alternate. Leaf alternate. We have seen leaf alternate is only 1, but opposite there are 2. Therefore, we will say leaf alternate, its name is this one, which is Bingolanaceae. While here now, because there are 2, we say go to 5. Now in 5, say 5a and B. This is simple because they are the only 2. Now this we will say, because there are 3, we'll say leaf trifoliate, and this one there are many, leaf pinnate. I will say leaf trifoliate, and then leaf pinnate. Now automatically you write the names where the leaf trifold is the compositae, the compositae, and the other one is papilionaceae. Now that's all. Thank you to our viewers. I'll urge you to subscribe, share and comment, and God will bless you. This work is not very easy, but do us a favor, please subscribe. Welcome. This is Kcse 2016 paper 3 biology, and let's see what you are supposed to do. In question 1, you are provided with specimen G. Now specimen G looked like this one, looked like this photograph. Now you are told cut off the petiole about 1.5 centimeter from the end where the leaf attaches to the stem. Then carefully make several thin cross sections through the piece obtained in A Roman 1 above using a sharp razor blade or the scalpel. Then put the sections obtained in water on a petri dish. Mount the thinnest section on a glass slide and add of iodine solution provided, and then observe the sections using a hand lens. Then draw a label diagram of the section observed. Now, as you can see this photograph, you should be able to know which class it is from. This it resembles a bean leaf. Now this is from class Dicotyledonnae. This will help you to draw the specimen you have observed using the hand lens. Now you are required to draw the specimen you have observed. Now the specimen looked like this. In a transverse section, it looked like this. This is a stem, transverse section of a dicotyledonous stem. And then vascular tissues are arranged. Then you are required to name. This is the epidermis. Then this is the cortex. Then here this is the pith, while this one is the vascular tissue. Now you should be able to note that don't specify like you write this is the xylem, this is the phloem, because you cannot able to see using a hand lens which is the xylem or the phloem. Now you write only the vascular tissue. This is a transverse section, of the dicoty. Let's see the longitudinal section. You should draw a specimen looking like this. And then here you draw a thin wall, the epidermis, both. Then you draw your vascular tissues. Now this is the epidermis. This is my cortex. This is the vascular tissue, the vascular bundle. while this one is the pith. This is a longitudinal section. Now this is from the dicotyledonous, and from a monocotyledonous, now you should draw for a transverse section which are scattered. You see, which are scattered, and in longitudinal you draw even here scattered many of these vascular tissue which are scattered. Thank you. B. Account for the following features of specimen G. Now you account why specimen G had extensive network of veins. Now this veins was to enhance the transportation of water and mineral salts. And why was specimen G have a tough leaf blade? This was to minimize the water loss. To minimize water loss, or to enhance support or prevent leaf from flooding. No, to prevent leaf from folding. Sorry, to prevent leaf from folding. Then strong and extended petiole. This is to offer support or to provide support. Now you have been told, state with a reason the class of plant from which the specimen was obtained. As you have seen, this is from class Dicotyledonae because has network veins, has the petiole, broad lamina, and etc. Now you should write this from class Dicotyledonae, and then reasons being has broad leaves, has broad laminar leaf, also has net veins, also there is presence of extended petiole, has the petiole, also has the pith, and etc. Then D, you are told, explain why the following procedures were necessary during the preparation of the section for the observations. Now putting the section in water on a petri dish. Now this one you should remember, when you put something you add water. This is to prevent desiccation or dehydration of the cell. Now you should write, this was to prevent desiccation or dehydration of the cell, prevent desiccation, and then using a sharp razor blade or a scalpel. This is to avoid distortion of the cell, to avoid distortion of the cell. Then here adding iodine solution to the section. This is to make it appear more distinct and clear. To appear, this is to appear or to be more visible, to appear more distinct and clear, or to enhance visibility. Why did we cut a thin section? For easy diffusion of iodine solution, or to allow penetration of light, to allow light to pass through, or for easy diffusion of iodine solution. Question 2. Study the photograph below and then you're required to answer the questions. Now, as you can observe this photograph, you can see there are two organisms. Now let's go straight to the question. State the type of the biotic relationship exhibited by the animals shown in the photograph. This, as you can see, this is the predator-prey relationship. Now we should write is the predation, because one organism killed the other in order to get food. So you can see this is a lizard and this is a snake. A snake is coiling on the lizard in order to feed on it. You can write predation or the predator-prey relationship. And then you are told, identify which of the two animals, E and F, have the least biomass. Now here you should be able to know biomass decreases as biomass decreases from the lowest trophic level to the highest trophic level. Now here, as you can see, the snake has the highest trophic level and the lizard has the lowest trophic level. So biomass will decrease from the lowest to the highest. Therefore, the animal that have least biomass is the snake, which is specimen E or animal E, the snake, because biomass decreases from the lowest to the highest. Now to give a reason, you will say animal E has the highest trophic level. Therefore, the biomass will decrease. Animal E having highest trophic level, therefore biomass will decrease from the lowest to the highest trophic level. That's the answer we're required to give. Now here you are told, explain the concept of survival for the fittest in relation to the organisms illustrated in the photograph. Now here, in an ecosystem consisting different organisms, one must have features that adapt to the environment. So you would say organisms that are in the same ecosystem compete on resources. First of all, organisms that are in the same ecosystem, they must compete on resources. When they have compete on resources, the organisms that is well adapted will survive. The organism that is well adapted will do what? Will survive, while the other one will be eliminated. Therefore here, E having more power, it kills F and feeds on it. Now here the key thing you are supposed to say that they must first of all compete on the resources, and the organism that is well adapted will survive while the other one will be eliminated. Therefore, then you say in our photograph, E will have high power to eliminate this one. Then you are told, explain 3 visible survival adaptive features for the organism illustrated in the photograph. You will say E is very long to coil around the prey. You see, yes, as you can see, the snake is very long to coil in the prey. And then you say both E and F, both animals camouflage to the environment. They camouflage with the environment. So you can see they camouflage with the environment. The environment here is brown, and both of them are color is brown. That is to camouflage. And then next, the snake, which is E, has more muscles or more muscular to attack, has more, it is strong, has more muscular to attack the prey. Lastly, the photograph below illustrates a procedure carried out to study gases exchange structure in a certain organism. Now as you can see, this is a fish, and this is a pair of concepts. This is a board. Then we are told, identify 2 dissecting tools being used in the procedure illustrated. This is the pair of concept. This is a pair of forceps. Spelling mistake. And then there is a dissecting board. These are the 2 dissecting board. So the tools used. As you can see, this is a fish. Name the class of the animal used. It's from class species. Then state any 2 visible characteristics from the photograph to support your answer. There is presence of scales, presence of gills, presence of fins, and etc. Then you are told, name the part of the organism labeled H and state its function. H is here. Now this is the operculum. Then the function of the operculum is to protect the delicate gill filament from the mechanical injury. You can say from mechanical injury. Also, this operculum opens and closes to facilitate inspiration. Another function, it opens and closes to facilitate inspiration and expiration. This is inhalation, exhalation, and expiration. Then D, you are told, draw the gaseous exchange structure under the study on it. Label the site for gaseous exchange. Now read the question carefully. Draw the gaseous exchange structure under the study on it, and then you label the site for gaseous exchange. You only label the site for gaseous exchange, that is the Gill filament. Now we draw our structure. Start our Gill bar. So our Gill bar, then our Gill filaments. Should be able to draw a nice diagram than mine in order to score good marks. These are the Gill filaments. Those are the Gill filaments. Then here I have my Gill rakers, like structures. Yes, this is my Gill rakers. Now you have been told label only the site for gaseous exchange. Now you label Gill filaments. See, I have pointed to, therefore should write Gill filaments. Then how is the part labeled in D Roman 1 adapted for efficient gaseous exchange? Now here you are being asked how Gill filament is adapted for gaseous exchange. Here you are being asked the characteristics of respiratory surfaces. Now the first one, it is moist. Now the Gill filament is moist in order to dissolve the respiratory gases. Yeah, it is moist. The other one, highly vascularized in order to maintain a steep concentration gradient. And then you can say they are numerous, or there are many. They are numerous to provide large surface area, a large surface area for gaseous exchange, Yeah, that's what you are required in 2016 to get all the marks. Now, thank you. And then I want to remind our viewers to subscribe and then share to your friends in order to gain more knowledge. Continue viewing our videos and God bless you.